Hello, dear colleagues. This is my seventh lecture. In this lecture, I would like to talk about in silico analysis of a single nucleotide polymorphism in diffenzene molecule. My name is Professor Sergei Shityakov. I'm a head of the chemoinformatics research group at the Infochemistry Scientific Center, the ITMO University. I would like to give you a brief introduction on the diffenzenes, what is the function of the diffenzenes, why they are important, and what is the purpose of these projects, why we are trying to analyze the single nucleotide polymorphism in these molecules. First of all, diffenzenes are the molecules which are extremely important for the innate immunity to combat different diseases. For instance, some bacteria infections. As you can see on the slide, if you have a bacteria in the human body, the diffenzene will attack the bacterial membrane and create some kind of a pores. These pores would impact the metabolism of the bacteria, eventually leading the bacteria to death state. And diffenzenes are molecules which are interacting with different cells within the human bodies and secreted by the different cells. And if these molecules are impaired by the mutation, that probably and most likely it would also affect the function of these very important molecules. Therefore, the use of the single nucleotide polymorphism analysis for the immune-related protein may be useful for the development of the new drugs and the treatment of various diseases such as the bacterial infection. At this point, some mutation might be neutral and some mutation might be harmful for the function of these molecules. Therefore, we are searching only for the mutations which are harmful for the function of these biological molecules, and we call these mutations non-synonymous mutations or non-synonymous SNPs. As a first step, we wanted to screen the database, in particular the SNP database from the open source, where we could find some non-synonymous SNPs within the diffenzene gene, or after that when the gene is expressed, then this mutation would also lead to an exchange in amino acid within the amino acid sequence of the protein, and therefore would significantly impair the function of the diffenzene to attack the bacterial cells. Here you can see that one could use some functions such as the proven scoring function or polyphen scoring function to find this non-synonymous SNP either within the diffenzene gene, diffenzene B1 gene, or within the protein structure. So we did this analysis, and we have found some particular SNPs which might be the most harmful for the structure, for the function of this molecule. Therefore, they're changing significantly the structure of the molecule, and after once the gene is expressed, this would lead to the impairment of the function of the particular protein. We analyzed these mutations by using the Monte Carlo algorithm to assess the protein stability, as shown on these graphs, and it is clearly seen that some mutations would also lead to an increase of the protein energy, which would suggest that the protein structure might be impaired by this mutation, and the stability is going to be decreased. The second step would be to analyze how this impaired structure within the defensine B1 molecule would also interfere with the interaction of PIP2 molecules within the bacterial membrane. And we did the molecular docking, which shows that once the structure of this protein mutated, the interaction of lipid protein molecule is also impaired. Next, we did the molecular dynamics simulation to understand how the SNPs would interfere with protein binding sites and how it would also interfere with the number of hydrogen bonds, for example. Once the protein lipid system is submerged into the aqueous environment. As you can see, the mutations would also decrease the number of hydrogen bonds within the protein lipid binding sites, which also suggest that this particular mutation is harmful for the function of the DEFB1 protein. And this is all in terms of the in silico design, in silico analysis of non-synonymous SLPs in a defensine molecule. Thank you for your attention.